morning, good afternoon, everyone. Thank you for the introduction. I would also like to thank the organizers for giving me the opportunity to present this work. Today, I will present the results of one particular study, which was a pan-cancer analysis of pre-diagnostic blood metabolite concentrations in the EPIC study. Blood metabolite levels can reflect both endogenous and exogenous processes that are possibly related to cancer development. And so there were a number of previous prospective metabolomic studies in cancer research that identified association between metabolites and cancer risk, as you can see from this selected list of references. But most of these studies focused on one particular cancer type at a time, or when they did consider several cancer types, they usually studied them separately. On the other hand, there are a number of cancer risk factors, including obesity, for example, that are established risk factors for multiple cancer types. And so it sounds reasonable to expect that some biological mechanisms and therefore some metabolites be linked to multiple cancers. And this intuition is actually supported by some results from the literature. And in particular, Kuhn and colleagues performed a pan-cancer analysis in the EPIC Heidelberg study, where they analyzed the association between metabolites and three cancer types, breast cancer, colorectal cancer, and prostate cancer. And what they observed, for example, for this particular phosphatidylcholine, was that the trend of the association across the different quantiles was very similar for the three cancer types they considered. And when they combined these three cancer types into what they called overall cancer, they, of course, also observed a similar trend and with an even more significant level of association. And they observed a similar phenomenon for this isophosphatidylcholine, although this time the association was inverse. And so from these results, well, it seems that indeed some metabolites might be related to cancer risk for multiple cancer types. And so we decided to expand on this work and to perform a pan-cancer analysis, but this time with a larger population, studying more cancer types and using a different and arguably refined methodology. And I will give some details about it in a minute. And so our overarching objective was to identify metabolites that could be associated with overall cancer. So in other words, metabolites that could be predictive of cancer for most or at least many cancer types. And also to identify metabolites that could be associated with specific cancer types only. So we used data from EPIC, the European Prospective Investigation into Cancer and Nutrition, which is a large cohort that included about 500,000 participants back in the 90s from 10 European countries. And in EPIC, blood samples were collected at inclusion. So, well, as in any prospective study, these blood samples correspond to pre-diagnostic blood samples, in particular for the cancer cases that were observed during the follow-up. And so in this analysis, we used the metabolomics data, or using the biocrates kit, that were successively collected in 8 cancer-specific matched case control studies nested within EPIC. So basically, we had one study for breast cancer, another one for colorectal cancer, endometrial cancer, gallbladder and biliary tract cancers, hepatocellular carcinoma, kidney cancer, as well as advanced and localized prostate cancers. And so overall, we analyzed data from about 6,000 matched case control pairs. And in this analysis, we focused on the 117 metabolites that were common to these 8 studies. Yeah, so maybe before going further, I just wanted to draw your attention on the sample sizes here. As you can see, it varied quite substantially from one study to another, and in particular, the sample sizes were rather small for the gallbladder and HCC studies. I will get back to this point in the discussion, but this is something that you may want to keep in mind. All right, so now I will give some details about the statistical method that we use. So the general idea was to pull the data from the different type-specific studies to leverage the possible commonalities across the different cancer types, as illustrated here in this example, where for one particular metabolite, the level of association with the different cancer types may be similar. On the other hand, of course, if we simply pull the data and ignore cancer types, then we could not identify type-specific association, as in the second toy example, where the metabolite is only associated with cancer type 3. So basically, to be in the position to leverage the possible commonalities across the different cancer types while still preserving their type specificities, what we did is that we pulled the data and we applied the idea of what is called the data shared lasso, which is an extension of the well-known lasso that essentially relies on this reparametrization of the log odds ratio that measures the association between metabolite J and cancer type K, and which is written as the sum of two terms here. So mu J is, can be seen as the overall association between cancer and metabolite J, while the second term here measures how the cancer type specific association for cancer type K varies or deviates around the overall association. So it's this second term here, which is a type specific deviation, is here to allow some heterogeneity in the associations between metabolite J and cancer across the different cancer types. And so once we have this decomposition, the data shared lasso is a simple L1 penalized approach, just as the lasso, which penalizes the absolute values of the different terms. And so basically, we want to penalize these terms first because for instance, we want to encourage the overall associations to be mostly zeros, because we kind of expect that only a few of the metabolites are indeed associated with cancer overall. And we also want to encourage the type-specific deviations to be zero, as we expect some level of homogeneity for the associations between any particular metabolite and cancer across the different cancer types. And so once we have estimates for these different quantities, then we can easily reconstruct the log odds ratio for metabolite J and cancer type K. So if we go back to our toy examples, in this first scenario where the metabolite has the same level of association across the different cancer types, then the data shared lasso should be in the position to identify that the overall association with cancer is non-zero for this particular metabolite, while the different type-specific deviations should be zero. And so in this particular setting, the data shared lasso would mimic the situation where we would simply pull the data and ignore cancer type, which is, of course, the optimal strategy in this case. And in the second example here, where the metabolite is only associated with cancer type 3, then the data shared lasso should be in the position to identify that there is no overall association with cancer, no type-specific deviation for cancer type 1 and cancer type 2, but non-zero type-specific deviation for cancer type 3. And so in this case, using the data shared lasso would be somehow equivalent to using only the data for cancer type 3 to study the association between metabolite J and cancer type 3, which is, again, the optimal strategy in this case. So we use the data shared lasso under conditional logistic regression models, as usual when analyzing data from nested case control studies. So this allows us to target mutually adjusted associations. So basically, when we study the association between, say, metabolite 1 and cancer, this association is adjusted for the other relevant metabolites. And here we also decided to adjust the models for BMI, as we considered that BMI could be an important confounder in the association between the metabolites and cancer risk. And our analyses were also controlled by design for the matching factors, which included age, gender, as well as country of origin. Something important to note, though, is that as most multivariate analysis, the results of the data shared lasso can be quite unstable and difficult to interpret in the presence of strongly correlated predictors or metabolites in our case. And as you may know, and as is illustrated on this figure here, some of the metabolites of the biocrates kits are indeed very strongly correlated with one another. So, for example, sphingomyelins are highly correlated to each other, and also, well, other phosphatidylcholines are strongly correlated, and we also have like some strong correlations among some amino acids or among some acylcarnitines. And so, prior to the application of the data shared lasso, we decided to group the most strongly correlated metabolites together. And so basically we perform a clustering of the 117 metabolites. And so here's a summary of the result that we obtained for this clustering step. So basically from the 117 metabolites, we ended up with 50 clusters. 17 of them were actually made of single metabolites that were left isolated by the clustering step. So for instance, this second cluster here is made of only one acylcarnitine. And for the remaining 33 clusters, what is interesting to note is that they were all made of metabolites of the same class. So for instance, this first cluster here was made of 2 acylcarnitines. This one here was made of 3 amino acids. This one here was made of 6 phosphatidylcholines, and this one here, for instance, was made of 6 sphingomyelins. Moreover, so once we have these clusters, what we can do is to construct what we call representatives of the clusters, which are simply defined as weighted averages of the metabolites that comprise that cluster. And what we observed here is that the correlation between the cluster representatives and each metabolite that comprised the cluster were all very large. So they were always larger than 0.83. And in general, they were even larger than 0.9. And so this is reassuring in a sense, because this suggests that, well, basically from a purely statistical point of view, the cluster representatives and the metabolites that comprise that cluster are loosely speaking indistinguishable, and so that it makes, I mean, it's fine to work with the cluster representatives rather than the original metabolites. All right, so now I can finally present the main results from our analysis. So here you have one figure which summarizes all the log odds ratios that we were able to identify for the 50 metabolites or cluster of metabolites and the 8 cancer types here. So first, I won't get I won't discuss all these associations in details, but we can focus on a few type-specific associations first. And so, for example, here I highlighted a few type-specific associations that were consistent with previous findings from cancer type specific analysis in EPIC. So, for example, this positive association between this cluster of acylcarnitine and breast cancer was already reported in the breast cancer study conducted in EPIC. Similarly, this inverse association between histidine and colorectal cancer risk was reported in the colorectal cancer study in EPIC. And for these sphingomyelins, we identified an inverse association with hepatocellular carcinoma and a positive association with endometrial cancer, again consistent with the cancer type specific studies in EPIC. But we also identified a few additional cancer type specific associations, and in particular, this positive association between arginine and colorectal is interesting because it only showed up in models, in mutually adjusted models, and in particular in models that were adjusted for glutamine and histidine. And so this is why maybe this association was not identified in the previous colorectal study in EPIC, because in this study they used a simpler approach based on univariate analysis, and so this identification here illustrates maybe some of the strengths of our analysis, which again was based on a multivariate approach. Moreover, and maybe more importantly, our analysis allowed the identification of several metabolites that showed a consistent association across different cancer types. So first, we were able to replicate some of the findings of the EPIC Heidelberg pan-cancer study that I mentioned earlier in the introduction. So for example, for this phosphatidylcholine, we indeed identified a positive association with breast cancer, colorectal cancer, and prostate cancers, as in this previous study, but our results also suggest that actually this phosphatidylcholine could be positively associated with all the other cancer types as well. And similarly, for this lysophosphatidylcholine, we observed a consistent inverse association with all cancer types, except maybe with localized prostate cancer, for which the association was either null or, I mean, borderline positive, if anything. We also identified other metabolites that showed a consistent association with across the different cancer types. In particular, we observed a consistent positive association between proline and most of the cancer types, except breast cancer. And well, this is consistent with some previous results in the literature, and in particular, very recently, proline was shown to be linked to tumor growth in a Drosophila model, and it was also shown to be associated with metastasis formation. So, well, let's say that our results confirm that proline could be a marker of interest. And another metabolite that could be of interest is glutamine, for which we observed like very consistent and inverse association with across the different cancer types. And again, our results were consistent in a sense with previous results in EPIC that already observed that this amino acid was inversely associated with colorectal cancer and hepatocellular carcinoma. But here we were in the position to show that, well, this amino acid could actually be inversely also associated with all the other cancer types. And actually this amino acid was also reported to be inversely associated with pancreatic cancer. So it could even be associated with other cancer types that we were not able to study in our analysis. So it could be really a marker of interest, for instance, for cancer prevention. All right, so to conclude, we performed a pan-cancer analysis on a quite large sample of pre-diagnostic metabolomics data. One particularity of our analysis, and maybe one strength of this analysis, was the use of the data shared lasso, which allowed us to have an optimal statistical power for the identification of associations with overall cancer, while still allowing us to identify type-specific associations. So I didn't get the chance to go through all of them, but we also performed a number of sensitivity analyses to check the robustness of our results. And so overall, we observed that our results were pretty robust. And so they were suggestive of possible shared metabolic pathways, although, of course, as usual, we have to acknowledge that our study suffers from a number of limitations. One of them is its limited power for the identification of heterogeneities or type-specific deviations, and in particular for some cancer types like gallbladder and biliary tract cancers or hepatocellular carcinoma, since as I already mentioned earlier, the sample sizes were quite small for these two cancers. And also, despite the clustering step that we applied, there were still some strong correlations among some of the metabolites or cluster of metabolites, and this, of course, hinders the unambiguous identification of the true metabolic predictors of cancer risk. And so, as usual, these results would need to be replicated in other studies. And to conclude, I just wanted to thank the EPIC PIs and the EPIC participants, as well as the many collaborators who worked on this project, and in particular, Marie Breuer, who is a PhD student working with me and who performed most of the statistical analysis in this project. So that's it for me. Thank you for your attention, and I'll be happy to take questions.